Welcome everyone. My goal today is to spend the next 25 minutes or so introducing you to our different cell isolation platforms and showing you how you can use our products for your particular HLA application, whether it's serology, flow cross-match, or chimerism testing. So what is cell separation? Well, cell separation is the isolation of a specific cell population from a mixture of cells. This population of interest can be isolated by either negative selection, where all your unwanted cells are targeted for depletion, or positive selection, where just your cells of interest are labeled with antibody. Unlike cell separation systems from other companies, our beads are not directly conjugated to antibodies. Instead, we use a more stable and flexible technology based on what we call a tetrameric antibody complex, or TAC. This indirect linking of your cell of interest to magnetic particles through the tetrameric antibody complex gives our reagents a number of advantages over reagents that use direct conjugation. Because this TAC complex is so stable, our reagents can be used at room temperature or at 4 degrees, and they're okay even if left out on the bench for several days. In addition, our reagents have a long shelf life if stored properly at 4 degrees, which ensures a constant performance of the product over time. So the real advantage for an HLA lab is that you'll never have to throw out expired reagents, and you'll have to validate your reagents less often. In a busy HLA lab, finding the right tools that facilitate accurate HLA testing is important. Our cell isolation systems are particularly suited to HLA labs as they're gentle on cells, cells are viable and have intact membranes, they're quick, most isolations take less than an hour to perform, they are easy, each kit comes with a detailed, easy-to-follow protocol, and most importantly, the isolated cells give clear, reliable test results. Cells isolated with our systems can be used in many different HLA applications, including serology, flow cross-match, and chimerism testing. And because I don't want you to just take my word for it, I'm going to pause now and then to give some real-life examples of HLA groups who are using our reagents to isolate cells for each of these different assays. Now at STEMCELL, we have two main cell isolation platforms. RosetteCep, which is our density-based cell separation system, and EasyCep-RoboCep, our immunomagnetic cell separation system. I'm going to begin with RosetteCep, our non-magnetic cell isolation system. Now, RosetteCep works on any sample that contains red blood cells, including whole blood, cord blood, and bone marrow. RosetteCep has several unique advantages in that it's a one-step procedure that doesn't require any specialized equipment. So you don't need magnets, you don't need columns. All you need for RosetteCep is a centrifuge. And because RosetteCep is a negative selection procedure, your cells of interest are not labeled with antibodies or particles and are immediately available for use in any downstream application. But more importantly, what our customers tell us they like about RosetteCep is that it's quick, it only adds 20 minutes to a standard FICOL spin, it decreases sample preparation time, and best of all, it's easy. How RosetteCep works is very simple. Your unwanted cells are cross-linked to red blood cells using our tetrameric antibody complexes. As you can see in this electron micrograph, what you end up with are rosettes of red cells with an unwanted cell in the middle. This essentially increases the density of the unwanted cell, which causes them to settle out with the red blood cells following density centrifugation. The rosette sep procedure itself is very straightforward. You add the rosette sep cocktail to your blood sample and let it sit for 20 minutes. You then dilute the blood and layer it over Ficol or your favorite density medium. You then perform a standard Ficol spin. Your unwanted cells will pellet with the red blood cells, while your unlabeled cells of interest can be collected at the interface. And these cells are immediately ready for use in any downstream application. We offer a variety of rosette sep cocktails that should be of interest to HLA labs, including cocktails to isolate different T-cell subsets such as CD3, CD4, and CD8 cells, cocktails to isolate B cells, total lymphocytes, lymphoid and myeloid populations, and many more. This slide shows an example of the purities obtained using our T and B rosette sep cocktails. The rosette sep T-cell enrichment cocktail typically gives purities in the 90 to 95% range, while B-cell purities are anywhere from 85 to 95%. So this is the type of excellent performance you can expect from a one-step system that's quick, easy to use, and doesn't require the use of a specialized magnet. One problem that I'm sure everyone in this audience has encountered is granulocyte contamination in a mononuclear cell sample prepared from older blood. As you know, granulocytes become less dense as they get older, which prevents them from spinning through the density medium during a regular Ficol spin. Now, what you might not know is that you can use rosette sep to solve this problem. By adding our special granulocyte depletion cocktail, you can increase the density of your older granulocytes by cross-linking them to red blood cells through our tetrameric antibody complex technology. You simply add the rosette sep granulocyte depletion cocktail to your blood and incubate it for 20 minutes, then perform a standard Ficol spin. The now dense granulocytes will pellet with your red blood cells, leaving your untouched mononuclear cells to be collected at the interface. The result is a clean mononuclear cell preparation that is free of granulocyte contamination. As I mentioned at the beginning of the webinar, I'm going to stop every now and then to give examples of how some HLA groups are using our products to isolate cells for HLA testing. My first example is a large transplant lab in the Midwestern United States. Now, if anyone is interested in the exact lab reference, please let us know and we'll be happy to provide you with some additional information. So this particular lab uses the rosette sep total lymphocyte, T, and B cell cocktails to isolate cells for flow crossmatch, CDC crossmatch, and serological testing. They estimated that it took 3 to 6 months to validate rosette sep for these different applications. They typically receive less than 10 mL of blood per patient, and they perform rosette sep in 15 mL conical tubes. What they like about rosette sep is that it has decreased their sample preparation time dramatically. and it's an easy system that all their staff can perform successfully. So hopefully I've convinced you that RosetteSep is ideal if you're working with samples that contain red blood cells. For those of you who work with other sample types, we offer a different system called EasySep. Now, EasySep is a bit more flexible than RosetteSep in that it can be used to isolate cells from many different types of samples, including PBMCs, frozen cells, lymph nodes, spleen, as well as from blood, bone marrow, and cord blood. EasySep is an immunomagnetic cell isolation system that doesn't use columns and gives cells a very high purity. The magnetic particles we use are small, and as a result, they don't clog the flow cytometer, and they're virtually invisible on the cell surface, as you can see in this electron micrograph of a B cell positively selected using our CD19 positive selection cocktail. During an EasySep isolation, target cells are specifically labeled with dextran-coated magnetic particles using one of our bispecific tetrameric antibody complexes. These complexes recognize both the dextran-coated particle and the target surface antigen. Magnetically labeled cells are then separated from unlabeled cells by simply placing the sample in a specially designed magnet. The EasySep procedure is very easy to follow. A cocktail of antibodies is added to the sample and incubated for either 10 or 15 minutes, depending on whether you are performing negative or positive selection. Magnetic particles are then added to the sample and incubated for 5 or 10 minutes. The sample is then topped up to a predetermined level and placed in one of our specially designed magnets. After 1 to 4 rounds of separation, your positively selected cells can be collected from the tube in the magnet, while your negatively selected cells are poured off into a separate tube. One unique EasySep application we have recently developed is a fast way to enrich for untouched cells directly from blood without the use of phycol. In this procedure, whole blood is pre-treated with a hetastarch agent called HETASEP, which essentially reduces the red blood cell burden of the sample prior to regular EasySep negative selection. Currently, this system can be used to isolate highly purified T cells, B cells, and total lymphocytes in less than an hour from start to finish. From a single 5 ml tube of blood, you typically get back 2 million total lymphocytes, or T cells, and up to 500,000 B cells. And as all of these cells are untouched, they can be used in all downstream HLA applications, such as flow crossmatch, CDC crossmatch, and serology. Many HLA labs embrace automation as it frees up tech time and standardizes results. If your lab performs any kind of high-throughput cell separation, you might want to consider RoboSep, our automated EasySep system. RoboSep is a pipetting robot that automates both the cell labeling and separation steps of cell isolation. It is a standalone instrument that's small enough to fit in most biosafety cabinets. It has a very easy-to-use graphical user interface and a lid that can be closed if you're working with samples that need to be kept sterile. RoboSep can process 4 independent samples simultaneously. And by independent samples, I mean in quadrant 1, you could isolate T cells from whole blood. In quadrant 2, you could isolate total lymphocytes from PBMCs, and so on. The rotating carousel has spaces for your reagent vials, so your cocktail and your magnetic particles, your sample tube, your negative fraction tube, your waste tube, and of course your separation tube, which is in the magnet. One of the best features of RoboSep is that each quadrant, and therefore each sample, uses a unique set of disposable tips. This essentially eliminates the potential for cross-contamination between precious patient samples. The RoboSep is very easy to use. You simply select a protocol from the list on the graphical user interface and load your sample and reagents into the carousel following the prompts from the machine. You then press the green Run button and walk away. Depending on the protocol, you return 30 to 60 minutes later and collect your separated cells. And these cells are immediately available for use in any downstream application. The main advantages of RoboSep for an HLA lab are that it saves technician time, as 4 samples can be processed in about an hour with only 5 minutes of hands-on time. It also avoids sample contamination, as each sample is handled with a unique set of disposable tips. And as all the steps are fully automated, there is minimal handling of potentially hazardous samples. One of the great features of RoboSep is that it can be used to perform sequential separations that allow you to isolate more than one cell type from a single sample. This is ideal for any type of sample that's in limiting supply, such as pediatric samples, where you only get 1 or 2 mils of blood, and you need to isolate a couple of different cell types from it. In the example I've shown here, we were able to isolate B cells, T cells, and myeloid cells from just 1 tube of blood, of 4 mils of blood. CD19 positive cells were isolated first, followed by CD3 positive cells, and finally myeloid cells. As you can see, the purities that we obtained for each cell type were above 95%, and we were able to get back enough cells that we could make DNA for chimerism testing. RoboSep is ideal for any kind of clinical lab that requires high throughput cell isolations. For this example, I'd like to highlight one of our very first and most faithful RoboSep users. This particular group isolates CD138 plasma cells from up to 8 patient samples every day. What they like about RoboSep is that it uses dedicated pipette tips for each sample, so there is no issue with cross-contamination. And because the process is fully automated, they can get other things done while the RoboSep is running. Now that I've introduced you to our cell separation platforms, I want to focus on how our kits can be used in different downstream HLA assays. The first application I want to discuss is serology. If you are performing serological typing or serological cross-match assays, you want a cell isolation system that is quick, efficient, cost-effective, and most importantly, gives clean cell preps that allow for simple scoring without ambiguity. Both our RosetteSep and EasySep kits meet these requirements. Both systems can be used to isolate highly purified T and B cells, and cells from either system show bright and uniform fluorescence under the microscope, which makes it easy to score your live from your dead cells. This slide shows cells isolated by RosetteSep, by EasySep, or by a couple of our competitors. In this first panel, cells isolated by RosetteSep look clean, with no visible particles on the surface. In the second panel, cells were isolated using our CD3 positive selection kit. As you can see, the beads we use for our positive selection are visible on the cell surface. However, they're small enough that they don't obscure the cells, as some of the other beads on the market tend to do. So the clean cell preps that you can get from RosetteSep or EasySep make scoring of your serological typing trays very easy to do. Another assay routinely performed in HLA labs is the flow crossmatch. Cells used in these assays must be clean, viable, and, most importantly, have low background fluorescence. Again, both RosetteSep and EasySep kits meet these criteria. As RosetteSep isolates untouched cells, background fluorescence is not an issue. The same is true for cells isolated using EasySep negative selection. Our T-cell positive selection kits has low background, as observed in this figure, the red line. However, we do recommend that customers use a negative selection approach when isolating B cells for flow crossmatch due to the higher background that is naturally observed with this cell type. The last application I want to talk about is chimerism analysis. Chimerism analysis is performed by many transplant labs to detect the presence of donor cells in patient peripheral blood as a way to monitor post-transplant outcome in the patient. For this type of analysis, DNA is typically made from isolated cell populations. Now, because of the sensitivity of the PCR and sequencing methods used to assess chimerism, even a few contaminating cells from a different sample can compromise the outcome of the assay. Therefore, cell isolation systems must give cells of high purity and must not allow for cross-contamination between samples. Again, both RosetteSep and EasySep meet this criteria. Many labs, like our specially formulated RosetteSep lymphoid and myeloid cocktails, while other groups like the high purity that can be achieved using EasySep positive selection. In addition, one of the benefits of our systems are that cells can be separated directly from blood or buffy coat samples, which can significantly reduce the sample preparation time. We have a large HLA lab in California who uses RosetteSep to isolate CD3, CD19, CD56, and CD34 positive cell subsets for chimerism testing. As they process many samples every day, they like the reliability of the machine and the fact that it doesn't require daily maintenance. Another factor that is important to them is that RoboSep minimizes specimen handling by the technologists. So in this case, RoboSep is the ideal cell separator to process high volume samples and to isolate highly purified cells. I want to finish off this webinar by mentioning a few features that I think make stem cell stand out in the field of cell separation. First of all, any of our kits can be customized so that you can get the exact kit that you want. For example, we have one customer who orders our Rosette Sep Total Lymphocyte Kit, but they get it with extra CD56 antibody in it to remove all the NK cells from the sample, as that's what's required for their downstream assay. In another case, a customer orders our standard formulations, but they get them in tubes with colored lids to make it easier for their staff to distinguish between the different cocktails. So really, whatever your wish, whatever your desire, we will do our best to try to accommodate it. And I think this speaks to the fact that we have an excellent customer service department and wonderful technical support team that will really go out of their way to answer your questions. And finally, we understand HLA and will do what we can to help you validate our kits in your system. If you would like any further information, please don't hesitate to check out our website at www.stemcell.com or to contact us at info@stemcell.com. Thank you very much for your time.